This is Chapter 14, Principles of Disease and Epidemiology, and I chose this cover picture. It's a representation of what's called the Broad Street Pump in London, and a famous epidemiologist actually was able to link a cholera outbreak to this water supply, to this water pump in London. Prior to this, it was generally believed that diseases, including cholera, were caused by things called miasmas, which basically means bad air. They thought like that foul smells from like decaying corpses or from garbage and sewer and things like that would make you sick. And they didn't actually realize that disease could be transmitted in contaminated water. So this chapter has a lot of terminology, as you will see on the study guide. It basically says to know the terminology, and that really means any bold word in the PowerPoint you would want to know. The good news is we kind of already know a lot of these, so it's not going to be maybe as difficult as it might seem. There are a lot of terms, but I think you're familiar with a lot of them already. Pathology is the study of disease. Etiology is the cause of a disease. So you can see that used as like the etiological agent of chickenpox is varicella zoster virus. So it's just kind of saying the same thing as saying the causative agent, the etiological agent, or the etiology. Pathogenesis is the development of disease. Infection is invasion or colonization of the body by pathogens. Disease is an abnormal state in which the body is not performing normal functions. And when we're talking about disease in microbiology especially, we are talking about diseases due to an infection. So, diabetes is a disease, cancer is a disease, right? But remember, we are talking about infectious diseases in this class. We've talked about the microbiome in quite a few chapters now. So again, in this chapter, it is brought up because, as we know, it is linked to our health for sure. It is linked to keeping us healthy from what we've seen in a lot of studies now, right? So I don't think there's anything new here on the slide that you need to know. This Human Microbiome Project is, maybe you heard of the Human Genome Project, that they actually finished it before the deadline that they were working on. That's where they sequenced the entire genome of humans. And then they started this Human Microbiome Project a few years ago that is still in the works, where they're trying to really pinpoint, although it's a huge project and I don't know that it's as easy as the Human Genome Project in actuality, but they're really trying to pinpoint, specific microbes that are part of our microbiome and try to figure out what benefits and what relationships they have with us and how they affect our health. And it's a huge project, but obviously it's been going on and there's been a lot of great discoveries that have come out of it already. So again, I think normal microbiota are those normal residents that are in and on our bodies. They do not normally cause disease, but they can cause disease to us sometimes, even our normal microbiota. For example, the E. coli, it's a good guy in our intestines, but you get that E. coli into your bladder, you get a bladder infection, UTI, urinary tract infection, right? So sometimes our normal microbiota can lead to infections if they get into the wrong places in our body, or in the cases of like super infections, if we take a broad spectrum antibiotic, we kill off some of our good guys, then some of our other ones that are still normal, and they belong there, but they're normally kept in check and kept in small quantities. Now some of those can overgrow and cause problems. Transient microbiota is referring to those that are not normal, meaning they're not permanent, but they may be with us for some time period of days, weeks, or months. turns out people who work in the medical field have more transient microbiota. I would venture to guess that school teachers, especially those of younger age children, maybe people that work in other very public environments like restaurants and malls and places that may experience a lot of crowds, Disneyland when it's open, things like that, they may also have more than normal transient microbiota, because the more people you're around, the more likely you can kind of pick up some organisms temporarily. Now, these transients, again, may not cause disease, but the person who picks them up and carries them transiently may be a source of infection for others, right? They may bring it home, and maybe they have someone immune compromised in the house, and now they may expose that person, or even just another stranger who may be susceptible for whatever reason, or that person themselves when they pick up that transient. Sometimes that transient may lead to infection in that person. So the distribution of the human microbiome in our bodies depends on many factors. There needs to be nutrients in that location for the microbes to have food. There needs to be physical and chemical factors that are going to allow them to stick around, to not create too much of a hostile environment for them. Again, our host defenses, of course, not creating a hostile environment for them. The different mechanical factors that we have that may prevent the microbes from taking up residence in those areas. But we do have a lot of areas that are very hospitable for microbes to live on, and we have other areas that are not so hospitable for microbes. Like our skin, yes, we do have some normal microbiota on our skin, but it does have some unique conditions, being that it's dry on those outer layers of the dead epithelial cells. It's acidic, it's salty, and so you only have microbes that are really suited for that environment to be there. But definitely our mucous membranes, especially of our respiratory tract, our oral cavity, nasal cavity, lots of normal microbiota there. Our intestines has a lot of normal microbiota. Our stomach, not so much, but you can find one main bacteria in our stomach called H. pylori. They're all just generic, though. They're not actually saying exactly what it is. They're just saying bacteria in the nasal epithelium, bacteria on the stomach, venturing to guess, though, that may be H. pylori there, bacteria in the small intestine. But if you are interested in what specific bacteria are found in what specific body regions, this table is a great one to look at. This is not something I would really test you on, because it is more detail than we need to know, but I find it very interesting. You can read and see, like, okay, the skin, these are the main microbes you find on the skin, and then read the comments and you'll just see a little bit of extra info kind of about that area of the body and how it's either inhibitory or welcoming to certain microbes. So, and it goes on for a couple slides there. We did watch a great video back in chapter 1 on the microbiome that, talked about how, babies start acquiring their normal microbiome at birth. And I think even in that video, they did mention that there is a difference depending on how the babies are born. And then, of course, all the interactions after the baby is born as well. If the baby is, raised up in too clean of an environment, that will affect their microbiome. Or if they take antibiotics a lot, that's going to affect their microbiome. So again, lots of interesting studies coming out and new discoveries about that. So switching topics slightly, it still has to do with our normal microbiome, but kind of looking at more detail of the benefits of it. So the relationship that our microbiome has with us is called symbiosis, right? It just means living together. And there's different types of symbiosis. Where is this slide? There we go. I'll come back to that other one. So it just means living together, and that can be peacefully or not peacefully, because even parasitism is a type of symbiosis. But in parasitism, the microbe is benefiting at the expense of us. And again, in general, our normal microbiome are not parasitic to us, but they can become parasitic to us in certain circumstances. Some of the examples I gave you earlier with overgrowth, or if they get into a different area where they don't belong. Or, of course, we do experience parasitism when we actually get an infection with any kind of microbe, not just normal microbiota. But our microbiota itself usually is either with a commensal type of relationship or a mutual type of relationship. In commensalism, the bacterium benefits and we are unaffected. And so the typical example of this has always been Staph epidermidis, that it benefits, it gets to live on our skin, gets nutrients, gets a place to live, and that there's not a benefit or harm to us. And that's commensalism. Mutualism, both organisms benefit. Our traditional example of mutualism is E. coli in our intestines. It provides vitamins and growth factors for us, plays roles in our digestion, and actually who knows what else, right? And there's lots of studies still going on, but those are the few things that we do know. It produces bacteriocins that can inhibit other microbes, possibly pathogenic microbes, from growing in our intestines. And again, the E. coli benefits because it gets a place to live, it gets nutrients. So that is mutualism. So some of our normal microbiota, though, again, can be opportunistic pathogens, including that E. coli. If it gets into the bladder, it's an opportunist, and especially because there's not a bunch of other bacteria there to compete with. So it's just going to go crazy and grow like crazy in your bladder and cause a bladder infection. Let's go back to this slide now. So I was just mentioning some of the beneficial ways that E. coli actually helps us out when it's in our intestines. It's there attached to our intestinal epithelia. It's, of course, taking up some nutrients. And that's actually beneficial to us because it can be then out competing other microbes that may want to come in and reside in that place who might not be as nice as E. coli. So the competition for nutrients, the bacteriocins that I mentioned, which is, comparable to the antibiosis, the production of antibiotics by microbes. Bacteriocins are antibacterial substances that can harm other microbes. So E. coli produces those in our intestines. Besides that, if E. coli does fermentation, which it's very good at in our intestines, fermenting different carbohydrates like lactose, which maybe some of us, including myself, are lactose intolerant, then the bacteria can actually do those fermentation jobs for us. Still not always the best effects. That's why you can get a lot of symptoms if you consume lactose and you're lactose intolerant, but microbes can help a little bit with those processes. But also during fermentation, microbes produce acid, and acids can be inhibitory to other microbes that might want to come and grow in that area. So that's the producing substances that are harmful to invading microbes and affecting pH and available oxygen. And so all of these things that our normal microbiota may do in that environment that they're growing in our body, those can protect us from infection by other microbes. And that whole process is referred to as microbial antagonism or competitive exclusion. Basically, our normal microbiota is being antagonistic to other microbes, possibly pathogens that may want to come into that same area. Our normal microbiota is being competitive and excluding those other microbes and not allowing them to come in and grow in that area. And that is a mutualism, right? That is how our normal microbiota who are doing that competitive exclusion or antagonism can actually benefit us. So here's the examples I was giving you when we were looking at commensalism, mutualism, and parasitism. And so again, commensalism was with staph epidermidis on the skin, mutualism with E. coli in the large intestine. Parasitism, there's tons of examples we could pull, but they've used influenza. Obviously, any infection, though, would be a parasitism. So here's a question about a normal microbiota and preventing overgrowth of a harmful microbe. So Koch's postulates. So we saw Koch in chapter 1. He was one of the famous scientists who was around during that golden age of microbiology. He came up with these postulates that basically are a way for scientists to show the specific microbial cause of a disease, because again, remember, at that time there was a whole lot of belief that diseases were due to, being punished by God or by those miasmas or by spirits or, just lots of different kind of wacky, what we think maybe now are wacky beliefs, but they were pretty standard beliefs back in the day. So Koch and some of the other scientists, though, that did start actually discovering microbial causes of disease, they needed a real scientific way, to prove this, right? That's the thing about science is, you need evidence and you need support. And so these postulates do that. And so they're just 4, somewhat simple steps that could be carried out by scientists to show that there was a specific microbial cause to a specific disease. So number 1, the same pathogen had to be present in every case of the disease. So if you had a bunch of cows that were sick, and this is the case with Koch because he studied anthrax in cattle, then you would have to find that same bacteria in every cow that had anthrax. And so what you had to do to show that was you had to isolate that pathogen in the laboratory and grow it in a pure culture, and then see it under the microscope and see it on the Petri plate and go, yes, that's the same bacteria, that's the same bacteria, that's the same bacteria, right, from all the sick animals. Then once that pathogen from the pure culture, was grown and confirmed to be the same, then you would take that and you would inoculate it into a healthy laboratory animal. And now that laboratory animal must also show that same disease, and you must isolate that same pathogen out of that animal. And if you were able to carry out those 4 steps, you were able to show that microbial cause of that disease. And here you can see it on a diagram, but I don't know why they put 5 numbers instead of just keeping it simple with the 4 postulates. But you can see microorganisms are isolated from a diseased or dead animal. They're grown in pure culture. They're, of course, going to be looked at under the microscope to go, okay, it's a gram-positive rod or whatever it may be. Then they're going to take these pure culture bacteria, inoculate them into a healthy animal, see that same disease now in this animal that they saw over here, re-isolate out bacteria from now this animal, and confirm that it is the same bacteria as was observed in the first animal, right? And again, if they can carry out those steps, they can then say, yes, this gram-positive rod, which we've named, Bacillus anthracis, is the cause of the disease, anthrax. So, Koch's postulates, again, are used to prove the cause of an infectious disease. There are some exceptions to Koch's postulates. Some pathogens can actually cause several different disease conditions. Some pathogens can only cause disease in humans, and so you don't have any laboratory animals to carry it out on. Some microbes have never been cultured, like Mycobacterium leprae that causes leprosy. We cannot grow that bacteria in the laboratory. So then you can't actually carry out Koch's postulates completely when there's some of these situations. but they still are able to kind of work around them. But ideally, you want to be able to carry out those postulates. So moving on, terms dealing with classifying infectious disease. Again, these are terms that you're probably familiar with, although I think we don't always use them to the proper definition. So symptoms are changes in body function that are felt by a patient, and signs are changes in body that can be measured or observed as the result of a disease. And so I think in the general use, we just clump everything into symptoms. We don't tend to distinguish between these two. But here, we're going to want to distinguish, okay? So what is a change in a body function that you can feel but is not measurable per se? So, like discomfort, what we call malaise, like you just feel sick. You just don't feel right. You just feel icky, right? So that's a symptom. A sore throat, even a headache. You can ask a patient to rate your pain from 1 to 10, but even that is really subjective because some people have a much higher threshold for pain than others do. So any kind of pain really falls under symptoms. Signs, things that can be measured. So we can measure fever, right? Temperature, any kind of fluids being expelled from the body, vomit, diarrhea, etc. You can measure the amount or the frequency. Same with coughing. You can measure the frequency of the cough. Swelling, you can measure, right? So those things are all going to be signs that are measurable. Syndrome is when you have certain diseases that really cause these pretty typical signs and symptoms together. Then it's called a syndrome. But not all diseases have that because a lot of times signs and symptoms will vary from patient to patient because a lot of the signs and symptoms that result really depends on your immune system and what your body decides to do in response to the disease. And you've probably noticed that if you've had a cold or a flu or something, run through your house, and you may have individuals in your household that get really, really sick and others who don't get as sick. And some people get this symptom and others didn't get that symptom. And there are some diseases that do tend to have very, common ones that do occur, but it's not always that case. Communicable disease is a disease that is spread from one host to another, and therefore contagious diseases are communicable, right? But not all communicable diseases are contagious, because contagious means a disease that is easily and rapidly spread from one host to another. So you can have a disease that's communicable. It can be spread from one host to another, but not easily, and therefore it's not considered contagious. And believe it or not, leprosy is communicable, but it's not really considered contagious. It's actually fairly hard to get leprosy. You need to have prolonged contact, and usually direct contact with body secretions from somebody with leprosy. So again, all contagious diseases are communicable, but not all communicable diseases are contagious. But most of them are. You think about any of the herpes, chicken pox, cold sores, Epstein-Barr, which causes mononucleosis, the flu, the common cold, measles. Almost any disease you can name that we know is contagious, they're communicable and contagious for the most part. Most infectious diseases do fall into both. It's pretty rare to have one that's, communicable and not contagious, but I gave you one example with leprosy. But what about infectious diseases that are non-communicable? So again, we're not talking about things like diabetes and cancer and things like that, because obviously those are non-communicable. We're talking about diseases due to an infection, but they're non-communicable, meaning they're not spread from a host, from one host to another. So I'll give you a minute to think, but most of the time in class, students don't tend to come up with these very easily. Okay, so a host, really, maybe we need to define. A host usually means a living animal or person, okay? The host is a living organism, though. So what diseases can you get, but it's from a non-living source? Well, some of the most common are from soil. So you can get tetanus, right? You can get tetanus if you get some kind of wound, a puncture wound. Most people think of a rusty nail and it's not the rust, it's dirt. It's dirty. The endospores of the bacterium Clostridium tetani are in dirt and soil, and so that wound can then get contaminated. If somebody has tetanus, can you get tetanus from them? No. So that's why it's non-communicable. You have to get tetanus from the soil source. Same with botulism, except now instead of soil, it's usually food, contaminated food. Somebody who has botulism is not contagious, does not give botulism to another person. That other person has to eat that same food that was contaminated and get botulism. Other examples would be those fungal diseases, which we've touched on maybe a little bit in our fungal lab in our chapter 12, but like the fungal diseases like that we have here, valley fever, or Ohio valley fever that we have over in the Midwest and East Coast. Those also come from soil, from inhaling fungal spores from the soil. And when someone else has those, you cannot get it from them. You can only get those diseases from inhaling the fungal spores from the soil. Okay, so those are examples of non-communicable disease.
Epidemiology is the study of where and when diseases occur and how they're transmitted. The knowledge obtained from epidemiology is of critical importance in learning how to treat and prevent various diseases.
Okay, I didn't know it was done. So here's some terms with dealing with occurrence of disease. So dealing with epidemiology, really. So incidence is the number of people who develop a disease during a particular time period. Prevalence is the number of people who develop a disease at a specified time, regardless of when it first appeared. So prevalence takes into account both old and new cases. So I bet right now you could probably look up this kind of information about COVID, or at any time we're having any kind of epidemic or pandemic, or even just something, well, AIDS is a pandemic as well, something that seems, it's constantly present in the population, like HIV, AIDS, or tuberculosis. You could go on the CDC website or the WHO website, and you can look up this information on a variety of diseases.
The prevalence of a disease is the number of cases, including both new cases and previously existing cases. The expected prevalence of a disease is the prevalence that's expected based on patterns established by past observations. The incidence of a disease is the number of new cases over some period of time. Let's see how epidemiologists use prevalence and incidence data to evaluate the status of a particular disease. A disease is sporadic in a region if only a few cases occur in that region. A disease is endemic in a region if it normally and continually occurs there at a fairly stable rate. A disease is epidemic in a region if it occurs there at a rate significantly higher than would be normally expected. A disease is pandemic if it's an epidemic on more than one continent at the same time. Now watch the incidence and prevalence data and see if you can recognize when an epidemic or pandemic occurs.
All right, so that animation just nicely went through these definitions. Sporadic, pretty straightforward, only occurs occasionally. Endemic, constantly present in a population. This is common cold. Common cold is endemic. It is constantly present in our population. Epidemic is one that is acquired by many people in a given area in a short time. We usually have epidemics of the flu every year. Measles was considered an epidemic fairly recently when we had the outbreaks associated with Disneyland and stuff. Whooping cough actually was an epidemic a few years ago as well when we started having a boom in cases. And of course, we know right now we are in a pandemic with COVID, and we're in a pandemic with HIV/AIDS. We do have pandemics with flus, usually every few years as well. We had the last one being the swine flu, the H1N1 pandemic. So again, lots of graphs of data can be accumulated and put together by our epidemiological agencies, mainly in the United States. That is our CDC, the Centers for Disease Control, but we also have the WHO, which is the World Health Organization, and NIH, which is National Institutes of Health. A lot of those different agencies will accumulate data and put it together in graphs like that one shown. Let's see, we're at almost 33 minutes here. I'm going to stop this video and pick up with part 2.